Hello, and welcome to the Introduction to BioCyc webinar series, Part 3, Pathways. My name is Alexander Shearer, speaking for the Bioinformatics Research Group at SRI International, and I'd like to welcome you back to our Intro to BioCyc webinar series. So far, in Part 1 of this series, we introduced you to the BioCyc website, its capabilities, and how to find things on the site. Then, using the example of arginine acquisition and biosynthesis, in Part 2 we introduced you to compound pages and transporters on a BioCyc website. Now, in Part 3, we're going to continue to follow arginine through E. coli and introduce pathway pages in BioCyc. When we ended Part 2 of this webinar series, we were on the L-arginine compound page, and we went down, like this, and clicked on Arginine Biosynthesis 1, which took us to the metabolic pathway of arginine biosynthesis in E. coli. So here we are. This is an EcoCyc metabolic pathway page, and this is representative of a BioCyc metabolic pathway page. They all look basically like this. It's arginine biosynthesis 1, and you may immediately be wondering, what's the 1 for? Well, in the Metacyc database, we have many variants and examples of metabolic pathways. So for example, there are 4 different arginine biosynthesis pathways at the moment in the Metacyc database. Since the Metacyc database is the basis for the generation of our automatically generated pathway genome databases, our Tier 3 collection, when you look through the BioCyc site as a whole, you can expect to see different organisms with arginine biosynthesis 1, 2, 3, and 4. E. coli, where it was initially characterized, gets arginine biosynthesis 1, but you can expect to see other variants of different pathways as you look through the collection. A BioCyc pathway page, as you can see, contains a fairly normal-looking representation of a metabolic pathway, with pathway intermediates represented in text, and then arrows representing the reactions that show the interconversion of those intermediates. Everything in the pathway diagram is clickable. As I mouse over things, you'll see a little tooltip pop up to tell me what I'm over. Obviously, in this case, it's a little redundant. When I go over the reaction, it's not, though, and it actually tells me this is a conversion, and it shows me its inputs and all of its outputs, including the ones that we can't see right now. One of the great things about a BioCyc metabolic pathway page is scalability. So for example, if we want to be able to see those side compounds that are also involved in the reactions, we can click More Detail once, and now we've zoomed in effectively. We can see, in addition to the pathway intermediates, the things that are converted throughout the pathway, we can see the side compounds that are also involved in these transformations. We also get to see, over here, the enzymes that carry out these reactions, the EC numbers for the reactions when available, and the genes that code for the enzymes carrying out those reactions. And if I were to zoom again one more time, then I would actually be able to see the structures of the molecules that are undergoing the primary transformations in the pathway. So here we can actually see the molecular structure change as we walk through the pathway. And of course I can also zoom in the other direction. So if I go and hit Less Detail once, I hit it again, and a third time, now we've zoomed all the way out and we get this sort of schematic representation of the metabolic pathway, abstracting out a lot of the reaction steps so we don't see the intermediates between L-glutamate and L-ornithine. What we can see at a glance, however, are things like the feedback regulation here from L-arginine, the pathway product, to the first step input into the pathway from L-glutamate. Now, these feedback regulatory things were present when we were looking at the reaction and the pathways on different scale, but they're much more pronounced in this sort of schematic zoomed all the way out view, so you can tune the pathway view in BioCyc to suit your needs. In addition, there are a number of other buttons up here at the top of the page. There's a cross-species comparison. I'm not going to go into that now. That's actually covered in our Omics and Comparative Genomics webinar and webinar series. But it basically lets you compare the presence or absence of this pathway and its components across all the organisms in the BioCyc collection. Then there's a Download Genes button, which will give you a list of genes involved in this pathway. And there's a BioPax format button, which will give you this pathway in BioPax format. And finally, up at the right, there is this thing, which is an evidence icon. So we'll notice evidence icons scattered throughout the BioCyc collection, we attach evidence icons to things like pathways, gene regulatory proteins, other proteins, and enzymatic reactions in their associations with proteins. And this indicates on what basis we assert that this thing exists. So for example, in this case, we're saying, and I'll go over this now, as you see from this pop-up, we're saying that the evidence for this pathway comes from experiments, which means from lab experiments. Someone actually did bench work that lets us believe this pathway exists. Other symbols you might see include a computer, which indicates something that's inferred based on a computational method. You might see a book, which indicates that it comes from a literature source, but we can't trace it down to a specific experiment. Those are used very rarely. And you might also see a person's face. That's also rarely used, but it indicates that a curator for a given database thinks that a thing is there or thinks an assertion is true. In general, you're going to see a lot of these experimental flasks when you look at the EcoCyc and Metacyc databases, because they are basically derived from the experimental literature. When you look through the Tier 3 collection, you'll see a whole lot of computers because, of course, those are computationally predicted databases. And if I were to click on this evidence code, as I'll do now, it will actually take me through to an explanation of the evidence and a link up here to our evidence ontology. And now I'll return to the pathway page.